Hi, everybody. Thanks for the opportunity to chat with you today. My name is Aaron Hartmann. I'm a researcher at Harvard University in the States. I'm a marine biologist generally, and I focus on coral reef ecology. And today I'm going to talk to you about marine metabolomics, so how we think about chemistry, biodiversity, and ecology on coral reefs. So I will start up my presentation here. So that we're all on the same page, when we're talking about metabolites, these are things that also can be called small molecules. You can think of them as primary and secondary small molecules. You may have heard those terms, or primary and secondary metabolites, natural products, or just chemicals. None of these groups are mutually exclusive, but they are all names commonly used to describe metabolites, and they are chemicals produced by biological organisms. They are usually, when we're talking about small molecules in particular, they're about 100 to 1,000 Daltons, and they tend to be lipid derivatives or lipid-based molecules, at least those that we focus on. And the metabolome, so going from metabolites to metabolome, is thinking about all the small molecules extracted from a sample, or all the metabolites extracted from a sample. I will use metabolites and small molecules pretty much interchangeably throughout this presentation, but remember, small molecules are just one subset of all metabolites produced. And small molecules are the source of many human drugs, so they are very valuable to society. So when we think about metabolomics as a field, we're of course talking about the metabolites themselves, but then we're thinking about the omics side of it, exploring their roles, relationships, and actions of the various types of molecules that make up a cell. And so in particular, I'm focusing on the relationships and actions of metabolites as my focal area. So to make sure we're all on the same page when we're thinking about how do we collect metabolomic data, we go out, we collect a tissue sample. So that could be a single coral, a single sponge, a single alga, or it could be, say, a mixed sample that we take from a community of organisms. We then choose a particular solvent to extract a class of molecules that we're interested in. So in my case, we're thinking about lipid-based molecules. And so we use a 70% methanol solvent to extract. And so we're picking up mostly lipids, but that are some that are slightly polar. So there's some polarity to those molecules. If we weren't targeting those, we would use something, say, like chloroform, which extracts non-polar lipids. Chloroform and non-polar lipids tend to be things that are used to, say, store energy in the marine environment. That's things like wax esters and triacylglycerols. But when we're thinking about metabolites that have ecological functions, say in defense or in immunity or in predation, those molecules tend to be slightly polar. And so we use methanol to pull them out. And so if you look on the right, number 5 is this notion that you've extracted your molecules. You then use something called an LC-MS-MS. So that's liquid chromatography, which spreads out your molecules, followed by 2 mass specs. So we call this tandem mass spectrometry. The first mass spec detects individual molecules that hit the detector, and it detects the entire molecule. So it quantifies the mass very, very accurately for one molecule, and also the abundance of that molecule. That molecule is then sent through again, and it is separated this time and hits the second mass spec. So all these different pieces or chunks of that molecule hits the second mass spec. And then what you get out of that is effectively a fingerprint of the molecule. And so that's what I'm showing up at the top in 7. So all of these peaks here are parts of a single molecule. They're just the different parts that were that the molecule broke apart into. And so you can really think of this as like the fingerprint of your molecule. And so here, what I'm showing you are a bunch of spectral fingerprints or spectra of molecules. And so what we then want to do is ask, what is the relationship between these molecules? How similar are they? How different are they? Because their similarities and differences relate to their functions. I should note too that in the marine environment, it's been estimated we know probably less than 1% of the molecules that we can, say, detect on a mass spec. Do we know what they are and what they do? And even in humans, that rate only goes up to about 2%. So we really don't know what most molecules do. So we need to find additional ways, additional tools to find information about them. Okay, so we've got all of these spectral fingerprints. And then what we do next, using something called GNPS, which was developed in Peter Dorostein's lab at UC San Diego on the west coast of the US, is something called molecular networking. And so what they do is they take two spectral fingerprints and they line them up to see how similar the two molecules are. So on the far left, we see three different molecules and their spectra. In the middle, I'm showing you two comparisons of two molecules. So in the top, you see spectra feature 1 going up, spectra feature 2 going down. So we call these mirror plots because they're mirrored. Every time you see a star, the same fragment is found in both molecules. And where you don't see a star, the same fragment is not found. And so we use this cosine score, a value between 0 and 1, to say how similar all these are these molecules. So in this case, they're about 87.7% similar. It's not quite a percentage, but you can sort of think of it that way. And then below, you see two molecules that are slightly more different. And I've highlighted a couple of peaks where they differ using arrows. And so the cosine score there is slightly lower, right, 0.760. But from that, we can then say, okay, above a certain cosine score, molecules are closely related to each other and should be grouped into a family or a single network. And usually we use a value like 0.75 or 0.8 cutoff in order to do that. So what I'm showing you on the far right are two networks of molecules, basically groups of related molecules. And the connections between molecules in the network suggest that they're very closely related. So two molecules that might be very far apart from one another, but in the same network, are about as different as two molecules can get and still be within the same family. So I'm going to shift gears a little bit, but it's going to sort of give you some real world grounding into why we would care about networking these related molecules, especially when we don't actually know what they do. We might not know what any of them do, or we might know what, say, one molecule in a family does. So, and that brings us to the story of Tectitethya crypta, which is a shallow water Caribbean sponge. It's often described as a drab shallow water Caribbean sponge that likes to cover itself in sediment. So you can see from looking at it and from the environment that it's in, that it's the type of environment you would often sort of walk over with little care for what you're stepping on as you're walking in, say, to go snorkeling or scuba diving. But don't be fooled, it's a pretty amazing critter. In the 1950s, scientists isolated and / discovered something called nucleosides, which are molecules produced by this sponge that interfere with viral DNA and RNA polymerase. So the result of that is that viruses their replication is slowed, right? So they're not able to make more gene copies, they're not able to reproduce. And so these nucleosides were used to develop antiviral and anti-cancer drugs. They were first described in the late 60s, and then in 87, they were approved for something called AZT. And I bring up the question of what pandemic did this treat? It treated the HIV/AIDS pandemic. It was one of the breakthrough drugs in the treatment of HIV/AIDS in the 80s and 90s. Moving on from that, in the 2000s, drug manufacturers started using these nucleosides to create hypothetical molecules based on the structure of those from T. crypta. Basically, they created these artificial families or artificial networks of molecules based on what we would think of like a similar backbone that all nucleosides had. And so with these families, they then synthesized and screened thousands of these hypothetical molecules. In other words, they produced them in the lab and then screened them against viruses to see whether they killed them or slowed their replication. And so they found new molecules that were turned into new drugs to treat things like SARS, MERS, and Ebola. And then one that's in the news right now is remdesivir, which is one of the few drugs that's been approved to treat COVID-19. So here you have it where This single sponge that you would probably never even notice, even if you were looking for it, is the source of molecules that have been used to treat two pandemics, and more than two pandemics, I should say, given SARS, MERS, and Ebola, are also in there with COVID-19 and HIV. So pretty incredible, and also something that was done using molecular networking and thinking about the relationships between different molecules. And so I think it's important now to say, when we're thinking about relationships between molecules, what does that mean exactly? Well, oftentimes it just means that there's a very small difference between them. So the molecules are identical except for, say, one chunk of a molecule or a chemical, say CHO or two hydrogens or something like that. And so we actually can figure out what those are based on what I'm showing in the bottom right, which these numbers relate to the mass differences between related molecules. So you have two molecules that differ by 2.016 mass units. And so what that correlates to or corresponds to are two hydrogens, so gaining or losing a double bond. So they only differ in having one fewer or one more double bond. So brings up this question of why sponges, just to bring in a little bit of ecology here, is that sponges don't have very strong defensive systems. They don't have a shell, they don't have stinging cells that they can use to defend themselves. So they produce a lot of compounds that help them to fight things off. They're also one of the oldest organisms, oldest metazoans on the planet. And so they've had a long time of fighting against things like bacteria and viruses. So evolution has had a long time to work in their case. But of course, they're just one example. This species is just one example, and there's loads more useful metabolites to be discovered. So that brings me to my work, with thinking about biogeography, or what molecules are where and why we find them in different places. So why would we care about this? Largely related to functions, thinking about finding new functions, how new functions evolve, new functions being things like fighting viruses or antibacterial properties or defensive properties or predative properties. I'm also fascinated by it because there are very few annotations. So metabolomics is really one of the next great unknowns in science. And most studies in metabolomics relate to a single species found in a single place. And that's often done because even in a single species, we can't identify very much. So if we're thinking about, say, large spatial scales or community level questions, it starts to get very complicated very quickly. And also thinking about metabolites as an ecosystem service, right? So once metabolites are identified, they can be synthesized in the lab. So they actually tend to have relatively low impact on ecosystems. It's not that we go out and harvest every single Tectitethya crypta and extract this molecule out. And so they can be relatively low impact, but they have very high value to society and very high value from an intellectual property perspective. And so for thinking about where we find these ecosystem services distributed, we want to know, where do we look for new drugs? And then also think about how communities, so say like a country that has coral reefs, how do they value their reefs based on the metabolomic potential there, but also thinking about ecological justice, and that is thinking about, say, who owns that intellectual property and who benefits from the money raised from selling it. So all really important questions, and all things that having a better understanding of the biogeography of small molecules can help us have a better handle on. So what colleagues and I did in a massive international effort was we sampled 71 coral reef communities from throughout the Indo-Pacific, from Aceh, Indonesia, in the west, all the way out to the Line Islands in the east, which spans basically from the Indian Ocean to the center of the Pacific Ocean. So we sampled communities using these things called ARMS, which are called autonomous reef monitoring structures. We put them out on a reef, anchor them down, leave them for a couple of years, and then bring them back. And the idea there is that they're passively accumulating things that live there. So things settle on them, and then we can, sample them off and see what's there using things like next-gen sequencing. So what we did is we went out and collected these ARMS, after 3 years, we separated all the plates of them, scraped everything off, literally put it into a blender, extracted molecules out, detected them on the tandem mass spec, and then got all of these clustered spectra out, right? So all of these fingerprints of the molecules. And in addition to that, we also did amplicon. So that is isolating individual genes from these, from a bulk DNA extraction, to then look at community diversity. And we did this with the 16S gene and the CO1 gene, 16S being a way to look at bacteria and CO1 being a way to look at eukaryotes. And then we carried out shotgun sequencing. So now we had an opportunity to look at metabolomic diversity, bacterial diversity, and eukaryotic diversity, all from the same samples and all taken from communities rather than individual species. Okay, so it's important now that I define a couple of terms. We're thinking about richness. When I might use the word richness, I'm talking about the number of molecules or the number of species. When I'm talking about structural diversity, I'm talking about differences in chemical structures between metabolites in a sample. And in this case, I'll be talking about the mean of a community. So what's the variation? What's the mean in that community in terms of the diversity of chemical structures? And then finally, before we dive into the data, it's also important to note that nature plays in these backbones too. Nature played on these backbones well before community or before computers played on these backbones. And so we're interested in metabolite diversity within families in nature. So how are families diversifying, families of molecules diversifying on coral reefs? And so what I'm showing you here on the left, I'm showing you the small molecules, individual molecules, and on the right, I'm showing molecular families. So that's on the Y, and on the X, we're looking at the number of ecoregions they were detected in. We sampled in 6 different ecoregions across the Indo-Pacific. So in the far left, what this is showing you is that most small molecules are only found in a single ecoregion, and it declines from there to where very few molecules are found in 3, 4, 5, or 6 ecoregions. In contrast to that, on the right, what this is showing you is that molecular families tend to either be endemic or ubiquitous. Endemic meaning they're only found in one ecoregion, ubiquitous meaning they're found in every single ecoregion. Which brings up the question of why is that the case? So the answer to that, as far as what we've seen, is that it has to do with their functions. So molecules involved in things like defense and in immunity were found in small families. The types of things we often call secondary metabolites were found in very small molecular families, and they were also highly endemic. So these molecules tended to be the ones that both the molecules themselves and the families tended to only be found in a single ecoregion. In contrast to that, molecules that are sort of the basic building blocks of cells for cell structure, housekeeping, were found in very large families, and the molecules and the families tended to be ubiquitously distributed. That is to say that they were found in every ecoregion. And so this, to us, explains why you have a lot of families that are found in one ecoregion, a lot of families that are found in all 6 ecoregions. And so the broader conclusion from this too is that chemical structural diversity is important, right? The diversity of structures can tell us something about the function of the molecules, even though we can't identify very many of them. I should note here, you do see identifications. And so it's these few identifications. We got about 20 out of almost 10,000 molecules. And that's how we're able to then come to these conclusions and then kind of extrapolate out from there. So our next question was sort of a broader thing, which is to ask, where do we find the greatest richness of metabolites and the greatest structural diversity of metabolites, knowing that both of these things are very important when we're thinking about sort of the diversity of ecologies, the diversity of functions, and also the potential for discovering new medicines. And so there's two key things to remember. So the first is that biological species are making these metabolites, right? And so this would suggest to us that metabolite richness and metabolite structural diversity is likely related to biological species richness, that is the number of biological species that are there, and it might also be related to phylogenetic divergence. That is to say, the greater spread of species you have across the tree of life, the greater diversity of functions they have, the greater diversity of metabolites they make. And so we wanted to address these two questions. And so before I dive into this, I'm going to define a couple of terms. The first is, again, richness. So when I say richness in terms of biological species, I'm talking about the number of species that were found in an arm. So that is in a single community. And we're talking about them as amplicon sequence variants, or ASVs. That is, a single sequence was identified, an individual unique sequence. And then secondly, when I talk about phylogenetic diversity in a moment, what we're thinking about is taking all those ASVs, lining them up as best we can, and then saying, how different are those sequences in terms of the nucleotides that are there? And so we can calculate the pairwise distance between every single ASV sequence. So we line two of them up at random, how similar are they, get a value for that, throw them out, do the next one, do the next one, do the next one. You can come up with then a mean or an average for an entire sample of what is the average difference between sequences. And the idea there is that that gives you a sense of phylogenetic diversity. The lower the mean value, the greater diversity of organisms you have across the tree of life. So, showing you the data, I am intentionally doing the thing you should never do, which is to throw a million graphs up all at once. And the point I'm doing that to make is that we found no relationships in any case between metabolites and biological species, whether they were eukaryotes or prokaryotes, whether you're looking at richness or structural diversity or phylogenetic diversity. There was just nothing that we could come up with in this context that would predict the richness of metabolites or the structural diversity of metabolites. And that was quite surprising. And so the conclusions that we had from this is that but metabolite richness and structural diversity is unrelated to biological species richness or phylogenetic divergence, which was shocking to us, actually, because that is a common assumption. It is almost spoken as if we wouldn't need to come up with data that more species will equal more metabolites. But when we tried to test this using one of the first tests using communities and across a large spatial scale, we did not see a trend. Also interesting is that most metabolites are endemic. That is, they're only found in one ecoregion or even in one site. And endemic metabolites are more likely to have ecological functions than non-endemic metabolites. And so it all suggests to us that we may actually be underestimating the value of reefs with low biological species diversity. To elaborate on that a little bit, you can imagine having an area like the Line Islands, which had relatively low richness of metabolites. But what that doesn't tell you is how much endemicity they have there. And we actually did find that even though they didn't have that many metabolites and they don't have that many biological species, there still was a high level of endemicity, that is uniqueness of their metabolites. And that uniqueness is what's important, right? If we're thinking about screening new molecules for drugs. And so just because they don't have a lot of species or metabolites doesn't necessarily mean their reefs are less valuable. And so that's a really important point here. So I will close there. There are literally hundreds of people to acknowledge for this project, but just to highlight a few, the amplicon work was done by Emma Ransom, Tita Kiani, and Aji Wayu. And the metabolomics work, as I mentioned, was done in Peter Dorostein's lab, in particular working with Robert Quinn, as well as some analysis help from Don Wei Huang and Jordan Casey. And the leads on this grant, which is an NSF, a large NSF PIRE grant, are Forrest Rower, Paul Barber, Alan Collins, Chris Meyer, and Nancy Knowlton, which was an incredible group of scientists to work with. And so with that, I will see all of you in the live Q&A, and thank you for listening.